All right, we're going to start now. Please have a seat. Thanks. So my name is Ali Gifford, and I welcome you to this symposium on large-scale visual neural datasets organized by Kendrick Kay and I. Before starting, I would like to thank the VSS organizers for making this event possible, and also you for joining. And I would also like to ask you to please take a few moments to fill an online questionnaire through the QR code on the screen. This questionnaire serves the double purpose of taking the community's opinions on some questions related to large-scale visual neural datasets, and it will also allow you to vote for the questions which will be addressed during the second part of this symposium, the panel discussion. And after filling the questionnaire, you will also get an invite link to join the computational neuroimaging server on Discord, where you can discuss topics related to fMRI, large-scale datasets, acquisition, and analyses. And also EEG, I just heard. Okay, I'm going to wait until all of you scan the QR code. It's going to be available later as well, so don't worry if you miss it now. Oh, it's also in the second slide, surprise. If we can zoom in. Can you zoom in with the phone? Hey, Daniel. Whoa. Okay, so the topic of the symposium is large-scale visual neural datasets. And as the name suggests, these are datasets which are larger than usual in at least one dimension, let it be subjects or trials or conditions, although this usual is not well defined. They involve visual stimulation, and they also involve neural recordings. And during the last few years, we witnessed a sharp increase in both the creation and usage of these large datasets, suggesting that the field of vision science is entering a new framework, a new research framework characterized by big open data. So then the goal of this symposium is to address this new research framework through a community event, which will be divided into two parts. The first part, we will have talks by 6 researchers who are either creators or users or both of large-scale visual neural datasets. And each of the talk will be 10 minutes, followed by 2 minutes Q&A for a total of 75 minutes across the 6 talks. And this will be followed by a panel discussion where you will, through the QR code, you will choose questions which will be addressed during this part of the symposium. And these questions could range from, should large-scale visual neural datasets prioritize more subjects, conditions, or repeats? Should they prioritize multimodal or purely visual stimulation? Should they prioritize more naturalistic stimuli or more controlled ones? How can large-scale datasets best sample the stimulus or task space? What are their major risks, dangers, limitations? Which type of large-scale datasets are missing? Or how can they promote theory formation? So please make sure to fill the QR code before the panel the questionnaire to the QR code before the panel discussion. And if you don't manage to do it now, it will also be present on the title slide of each of the 6 talks. So with this, I wish you a good symposium, and Kendrick will now moderate the entire event, and we'll start with the talks.
Hi, everyone. Yeah, I'll be moderating. I'll introduce each of the speakers. These are fairly rapid talks, and I'll keep it on time. If there's time, quick questions, and we'll leave plenty of time at the end for the panel discussion / Q&A. So the first speaker to lead things off is Elena Coopers with a talk entitled The Natural Scenes Dataset, Lessons Learned and What's Next.
The Natural Scenes Dataset, or NSD, is a massive 70 fMRI initiative to bridge the neuroscience and artificial intelligence fields. 8 participants were scanned up to 40 times over the course of a year, and each participant saw up to 10,000 complex natural scenes from the Microsoft COCO dataset while doing a continuous recognition task. So they would fixate on the red dot and press the button to indicate whether they saw the image before or not. In addition to this multi-session fMRI experiment, NSD also included different types of scan contrasts and many extra measures, such as the object category localizer, retentopy, and resting state. NSD has made a big impact on the neuroscience field. Since its release in 2021, it has been used in more than 90 papers and preprints, and this list is still growing. So why is it so impactful? And what can we learn about this initiative when we create future large-scale data sets? Today, I will share the lessons learned from the NSD team, as well as the experience from users. Then I will give you a teaser of what's on the horizon, including an intracranial EEG dataset that uses the same NSD images as the fMRI subjects, and the next large-scale fMRI dataset initiative that we are currently working on. So what is prioritized in NSD? The NSD team aimed for a big dataset, one that could be used to train state-of-the-art machine learning algorithms. They collected a lot of conditions per subject, which is also known as deep or precision fMRI. Compared to other datasets, it had still a reasonably large number of subjects. So here I'm plotting the number of subjects on the y-axis against the hours each subject was scanned, and each dot is a publicly available large-scale dataset, and NSD is in the lower right corner. The second priority is to maximize general utility and flexibility. So NSD is designed to be a resource where the data supports a large hypothesis space. So big is necessary, but not sufficient. The team prioritized high SNR and high resolution using 7 Tesla MRI. And then a unique aspect of NSD is the comprehensive and extensive quality control that was published alongside the data. They basically made every trial count. They also pushed the envelope on acquisition and analysis methods because the off-the-shelf methods were not sufficient or not always sufficient. So, for example, they developed GLM single, which we'll also hear later about maybe from Jacob Prince, which was a method to accurately estimate single trial betas. And the creators or the team was paranoid on details and documentation. So if you see the NSD data manual, it's basically a second paper. So one example of optimizing the dataset is screening for the best subjects, as there is huge variability between the bold signal strength between people. So here I'm showing the 14 subjects that were initially tested or screened, and each column is a scan on the object category functional localizer, and the color map is variance explained by the images. So brighter color here is better. And based on this localizer, they excluded 6 subjects, so 14 became 8, because they didn't want to waste any scan time. And the screening worked. Here I plot the subject ranking. So the yellow dots are those subjects that were actually in the NSD dataset, and they are plotted against the noise ceiling on the NSD experiment, which is the variability amongst the image repeats, which has a nice linear fit. And when we extrapolate that to those subjects that were excluded, that would actually, or this would suggest that including those subjects would have resulted in more, but actually poor data quality. Subjects could also see performance for every session and got bonuses if they did well. To understand the user experience, we are currently running an anonymous online survey, and these results are preliminary because you, all of you, can still participate in this survey. You can find the link on the NSD website or this QR code. We advertised widely, and our current sample is mostly US and Europe based, mostly academics that really like cognitive neuroscience and study vision. I guess this image doesn't work so well. And half of these people that we have so far at least used NSD once. So when we asked what purpose did NSD serve, when they used it, most of the people said they used it for running exploratory analyses, but not exclusively. So they also tested it or used it for testing specific hypotheses and many other things, including making art. So I would say general purpose priority was definitely working out in practice. When we asked users about how they felt about the ability to answer their research question, they said they were somewhat or extremely satisfied. They really appreciated the quality of the data as well as the documentation. And one thing that stood out was the availability of the fully pre-processed data, so these single trial beta values. People were less fond about the choice of the MS-COCO image dataset due to the lack of diversity And people wanted more controlled stimuli or dynamic stimuli, and they wanted more behavioral tasks or data that probed cognitive processes, as they found it difficult to establish brain behavior correlations. So what's next? First, I would like to introduce NSD intracranial EEG, which is an amazing dataset effort led by Dr. Dora Hermes and the Multimodal Neuroimaging Lab at the Mayo Clinic in Rochester, Minnesota. So here, patients with intractable epilepsy are monitored in the hospital, and they have these stereo EEG electrodes. So they viewed the 1,000 common images shared across the fMRI subjects. So here's an example patient brain, and each electrode has multiple contacts. So both cortical and subcortical contacts, if you're interested in the amygdala or hippocampus. And the colors show the different visual areas from anatomical atlases. Across the patients, they have more than 300 electrodes showing stimulus selectivity, and over 90 are confirmed to be in retinotopic areas. And I want to point out that this visual coverage, or this amount of visual coverage, is rare and is very exciting about this data set. Another exciting aspect is the concurrent high-quality eye tracking. So subjects were doing the same, or patients did the same task, and were instructed to fixate. In this case, they did well, but sometimes they made eye movements. And if you want to correct for that, you can, because there's high temporal precision in these eye tracking data. Design was optimized for intracranial EEG, so stimuli are on and off for 800 milliseconds. And here I show an example view on electrode time series and one for LO, which is a high-level visual area. And we can see very nice features, so evoked potentials when the stimuli came on, or when the stimulus was taken off the screen, as well as alpha oscillations during rest. So these are single trials, so this is extremely clean data. In the spectral domain, you can see time-varying broadband, narrowband gamma, and alpha power changes. So these rich temporal features can give complementary insights to the fMRI data. The Mayo Clinic will wrap up the data collection by the end of the year, and the data release will include the raw, pre-processed, and all the files that you want, eye tracking, behavior, you name it, all in standardized BIST format. And if you want to learn more about this data set, I would recommend going to Harvey's poster tomorrow or Zishan's talk on Wednesday. So we're also planning the next large-scale data set effort at Minnesota. And VCD, the visual cognition data set, will have all the good stuff you want from NSD. Don't worry, we'll make it happen. But the exciting part is that we're going to prioritize two aspects that people thought were missing that we learned from the survey. First, diverse stimuli, and then diverse tasks. So we're going to have both carefully controlled artificial stimuli, such as Gabor's, and these random dot motion stimuli, as well as faces and words and naturalistic scenes. In addition, we're going to sample many different types of tasks on a common set of stimuli, and this will enable brain behavior correlations. And here we're going to use these very well-accepted tasks, such as orientation or motion direction discrimination, as well as working memory and other tasks. So the takeaways, in sum, vital to NSD's impact was the intention for it to be a community-oriented, broadly scoped data set with the purpose of being a publicly available resource, and the maximization of quality and quantity, basically optimizing all aspects of the dataset creation, having attention to details, frequent quality control, and extensive documentation. Keep an eye out for exciting new datasets, so NSD IEEG and the Visual Cognition Dataset, or VCD. And then I want to end with this open question: what is the future of big data neuroscience? Because this is an exciting time, but it also raises interesting questions. For example, and we're actually writing a perspective piece on this, which we just put online, so if you're interested, you can read it here. So we're thinking about these issues, like how do we integrate big data sets? How do we avoid collision? Like, we don't know who is using these data sets or not, and we don't know if people are creating the same data sets on the side, or like in parallel. How can we build an inclusive community of big data sets of creators and users? How can we learn from one another using the same data set? And funding, because the small data sets are important and we should keep funding them. So how do we create an ecosystem where there's both big and small data sets, because they both have value and impacts. And with that, I would like to thank you, the NSD team, the VCD team, the Mayo Clinic team, CVN Lab, our funding sources, and thank you for the symposium organizers for inviting me.
We have time for a question or two, and you can also ask Alina questions at the end of the panel discussion. But are there any questions? Feel free to just shout it out. I can hear. Questions, comments? Ali?
I was wondering how many subjects and how big is it going to be, this new data set you're collecting? Is it comparable to NSD? Is it bigger, smaller?
It's going to be same NSD style, probably 8 to 10 subjects scanned over a year, so 30 to 40 sessions. Yeah.
Okay, so around also more or less 30,000 trials? Was it something like that? 40 sessions, yeah.
Well, the trial structure is going to be different, but yeah, it's going to be big, ambitious, bold. Yeah, come prepared.
Okay, let's give Elena another round of applause. All right, our next speaker is Wonmuk Shim, and the title of her talk is Exploring Naturalistic Vision in Action with the 17 Naturalistic Perception, Action, and Cognition, or NatPac, dataset.
Thanks. All right, thank you, Kendrick. So there have been many attempts to build large-scale neuroimaging datasets, including ones that are more focused on vision, which is the main theme of this symposium. So these large-scale human neuroimaging datasets have played a crucial role in mapping brain and cognitive functions at a macroscopic level. You have already seen this figure. So when we plot existing large fMRI datasets along two main axes, the number of subjects and the hours of fMRI data per subject, we can see two extremes. So limited sampling from many subjects on one axis, like the UK Biobank and HCP, like we already know, and extensive sampling from a few subjects on the other axis, like senior mod and NSD, which was nicely introduced by Alina in the previous talk. So if we had unlimited resources and time, we would want to aim there, the ideal, but disregarding this ideal, notice that even around there, there's not much of a data set available. So in our recent large-scale data collection effort, the 70 naturalistic perception, action, and cognition NetPack dataset, we aim for that point. So extensive sampling for complete models of individual brains, about 12 to 16 two-hour sessions per subject, and a not so small number of subjects, of about 100 subjects, planned to be scanned over 5 years. That can still give us information about individual differences. So in this 70 NetPack projects, we have three main goals. To provide, first, large-scale, high-spatial resolution, whole brain anatomical and functional imaging datasets obtained at 70, along with behavioral and physiological data. And second, extensive coverage of sensory, cognitive, and affective processes within individual subjects with a wide array of structured and naturalistic tasks. And lastly, new naturalistic tasks designed to probe many of the higher order cognitive functions, including watching narrative movies, free speaking, and playing interactive 3D video games. And this is the status of our data collection. So we started collecting the data in the fall of 2022, and as of now, 11 subjects have completed 12 to 16 sessions. Unfortunately, one subject dropped out of the study. And we are currently in the process of scanning 4 new subjects. To give you some idea about the quality of our data set, here are the results of the first pass data quality check. So both temporal SNR and mean FD, meaning motion, appear to be within reasonable ranges, although lower TSNR and higher motion were found for naturalistic tasks compared to structured ones, given the dynamic nature of speaking and interactive gameplay. So now on to our tasks. So as I mentioned in a previous slide, we use tasks encompassing a broad spectrum of human cognitive functions, from low-level unimodal sensory and motor processes to high-level transmodal cognitive ones. For example, we included more than 13 structured tasks to probe individual cognitive functions, ranging from vision, pain, action, and attention, memory, and progressing up to the hierarchy to language, social cognition, and emotion. And like you can expect, we identified many of the well-established functional areas involved in a wide range of processes, from sensory to motion, from sensory to motor functions, and also encompassing many cognitive functions. So most functional areas were consistently localized across different subjects, although there are some individual differences. Crucially, in addition to these structured tasks, we included 3 naturalistic tasks, which were designed not only to assess individual cognitive functions, but also to cover many of them simultaneously in an integrative manner. So for example, while watching movies, we engage most perceptual, linguistic, and social functions. When we freely speak our thoughts and memories, maybe sensory processing plays just a minor role, but there is an intense use of language, reasoning, memory, and social functions. Lastly, when we're actively interacting with environments and pursuing behavior goals, we engage in nearly all the perceptual and cognitive functions listed here. So before moving on, I'd like to mention a couple of things why we chose to include naturalistic tasks in our study. So many researchers, including myself, typically employ a reductionist approach. So one of the underlying assumptions here is that we can probe each elementary cognitive functions in isolation using simplified and well-controlled stimuli, and maybe put them together later for a more comprehensive understanding of more complex cognitive processes. But we may not be able to guarantee that these reduced stimuli indeed preserve the essence of cognition in the real world. Also, in a typical laboratory experiment, because identical and independent trials are repeated many, many times, the interaction between the agent and environment is just not there. So in contrast, in real-world contexts, we explore the environment while interacting with other biological and social agents and making hierarchical decisions, plans to achieve their own behavioral goals, and also adaptively updating them. So to simulate this process in a naturalistic setting, we developed a new set of interactive 3D video game using Minecraft as a platform. And this is our experimental setup. So using an MR-compatible game controller and an eye tracking system, we collected a rich set of gameplay data, fMRI scans, and eye tracking information while participants engaged in the game. Our participants played a variety of games, such as gathering fruit in an orchard, virtual orchard, based on their beliefs about environmental regularities, or herding sheep through hierarchical strategic planning, and forming relationships with in-game characters while navigating a dynamic social world. So each of these tasks is designed to tap into a distinct set of cognitive functions. And all these tasks have hierarchical structures consisting of a primary goal, multiple sub-goals, and also subsequent action sequences and elementary actions. In today's talk, I'm going to focus on the second task, shepherding. So in this task, participants are required to continuously make plans and execute actions to herd as many sheep as possible to a specified location, while avoiding the risk of losing sheep due to falling into puddles or being attacked by a randomly spawned fox. So to implement more realistic collective behaviors of the sheep and herding in our task, we adopted a computational model from a previous study that were developed based on GPS data collected from a real flock of sheep and a shepherding dog. Okay. So this task consists of 3 stages. First, initial planning of the best route by examining the map. And second, restructuring the environment by breaking fences to make shortcuts or building new ones to protect the sheep from the puddles. And third, the actual herding process during which they navigate the environment and guide the sheep to a designated area. Okay. To analyze this naturalistic task as it employs free viewing, it is essential to obtain a high-precision eye-tracking data that allows us to identify which object the participants are looking at each moment during the gameplay. So in this task and other visual experiments in our dataset, we obtained the camera-based eye-tracking data, but gaze accuracy can significantly drop when the participants move their heads just a little bit from the calibrated position. So even a 0.5 millimeter head motion can lead to 3-degree errors in eye tracking. So to mitigate this problem, we have developed an algorithm, motion-corrected eye tracking, we call MoSet, which estimates motion-induced drifts using motion parameters obtained from EPI pre-processing pipelines and regresses them out. This procedure has proven to be highly effective, and we are currently preparing a toolbox for its broader use. And you can hear more details about this method in the poster session on Wednesday morning. Okay, there are many, many interesting aspects you can look into in this task. But since this is a VSS, among them, I picked one, which is how potential reward loss modulates visual representations. Okay, using a voxel-wise PRF model from a conventional PRF experiment and predicted bold signals based on gaze-centered visual input from high-precision gaze estimates provided by the MoSet, we removed the retinotopic visual effects from the data and applied an encoding model to the residual to find spatially specific reward responses in the visual areas. So we found responses to reward loss, meaning losing the ship to puddles or a fox in the visual cortex, specifically tuned to the location where the reward loss occurred. And these responses increased as the time approached the onset of reward loss. And interestingly, this pattern was not observed for near loss events in which the ship approached the puddle but did not really fall into it, so indicating a distinction between actual and potential losses. So moreover, this reward responses was correlated with the successful loss avoidant behaviors, which are actions to save the endangered ship. So we think this study illustrates the interplay of perception, action, and cognition as an integrated process in naturalistic settings, which is rarely studied in conventional laboratory paradigms. So again, you can find more details about this study at the Tuesday afternoon poster session. So as large data set, whether it's a wide or broad or a deep, can be useful in addressing many of the cognitive neuroscience questions. We hope to contribute high quality data sets with comprehensive and dense sampling across a wide spectrum of the cognitive landscape. And since each component of our data set can be separately analyzed in many different ways and also in a combined fashion, we hope to provide some level of, high level of flexibility in the researchers' data analysis. And okay, I'd like to acknowledge the entire team behind this project. Their efforts has been absolutely crucial. And I want to give a special shout out to Kendrick, who suggested the current name of our dataset, NetPack. So thank you for listening.
We have time for a question or two. Oh, yeah, Layla.
Okay, thanks. This looks so cool. Can I ask how long the movie is, and in more generally, how you think about how to weigh the relative durations of data you get for these different tasks?
Let's see. So the movie, I think we used the short film, so like a 20 to 30 minutes movie each. So we have 4 movies here, and also for the Minecraft-based interactive video games, we had a whole session or 2 devoted to each of those tasks, whole 2-hour session. So it's 2 hours, either 2 hours or 4 hours per Minecraft-based task. So that's rough estimates.
Okay, if there's further questions, we can hold them also to the panel discussion. Let's thank Won Muk again. Our next talk will be given by Yann Charest from the University of Montreal. His title is Exploiting Large-Scale Neuroimaging Datasets to Reveal Novel Insights in Vision Science.
Okay. Hello, VSS. I'm happy to be here today to talk in this symposium about a few insights that we can gain from large-scale data sets and how they have enabled novel findings about brain function in vision research. So the human brain extracts complex information from visual inputs, including objects like the kayaks, the sail, their spatial and semantic interrelations, and the interactions with the environment, like the windsurfer navigating this lake. However, coming up with a computational model or quantitative approach for studying this information can be challenging. So in the next few slides, I'll show you a set of results kind of telling us how contextual information encoded in large language models, yes, language models, can be beneficial for modeling how the brain processes information about visual scenes. Large language models are trained on massive data sets of textual tokens with billions of text tokens, and the scale of this textual information that they ingest can help them converge decently to a representation that characterizes the structure of the world. So we're going to ask the bold question, can large language models, and especially embeddings of scene captions in these large language models, be an effective way to capture the rich information conveyed by visual scenes? So cognitive computational neuroscience is undergoing a transition similar to what happened in artificial intelligence over the last decade or so. And this has helped generate novel insights about the human brain. With its high-resolution brain responses to over 70,000 natural scenes, the natural scenes dataset has all the right ingredients, including captions, scene captions, paired with every single scene presented. So it has all the right ingredients to test novel hypotheses linking visual representations in the brain with large language model representations. To test whether the representational format on which LLMs converge can be useful to make predictions about brain responses to visual scenes, we will use two complementary approaches. Each stimulus is paired with brain activity from the NSD dataset and scene captions that came with the Microsoft COCO dataset on which NSD was sampled the stimuli. The scene captions were passed through a large language model, and we used MPNet, which is a state-of-the-art large language model trained on sentence embeddings. To find out whether there's a link between these embeddings of scene captions and fMRI responses to the natural visual scenes, we will use representational similarity analysis and voxel-wise encoding modeling, which I will show you in the next few slides. So I'll start with our RSA results. To devise a model based on the scene captions passed through MPNet, we compute the representational distance between their respective embeddings. We place this distance in a representational dissimilarity matrix, or RDM, and we repeat this process across all pairs of scene captions embeddings, and this gives us a model that is entirely based on textual input, on scene captions of the visual stimuli. To see whether the MPNet embeddings can predict brain responses related to the visual scenes, we will use RSA with Searchlight. We will compare the RDMs constructed from the pairwise activity patterns from seeing the visual scenes, and we will compare this to our model RDM that is based on the MPNet embeddings of the scene captions. This cortical flat map, which was done by cutting the cortical surface and projecting it on a 2D plane, shows where in the brain there's a significant representational alignment between the brain responses to the visual scenes and the embeddings of the scene captions. So we can see that in most of the brain regions, there's a significant correlation between the model and the brain, and that this peaks in high-level visual areas. This searchlight result is extremely robust, and we saw it in every single participant of the NSD dataset. Our results indicating that the brain representations are well characterized by LLM-like representations lead us to the intriguing hypothesis that LLM embeddings may be a powerful target for training visual models. So we trained recurrent convolutional neural networks to predict LLM embeddings of the captions associated with the input scenes. The model receives new image, raw image inputs of the natural scenes, and via nonlinear transformations and backpropagation, it tries to predict the embeddings of the scene captions that are associated with the images. We then use Searchlight RSA to quantify the representational alignment between our model and the brain responses from the NSD. So the Searchlight RSA result revealed a very similar network of brain regions, but with improved performance compared to the RSA Searchlight result we observed when looking at the LLM embeddings that our model was actually trained to predict. Again, this was observed in every single subject of the NSD dataset. We also ran a highly controlled model comparison, contrasting our LLM-trained RCNNs with RCNNs trained to predict multi-hot category labels, which are available with the COCO dataset. So these two models have exactly the same input architecture, the same input, the same architecture, but two very different task objectives. We also made sure that the training images did not overlap with the natural scenes dataset to avoid any kind of overfitting. To adjudicate between the two models, we contrasted their representational alignment, again using Searchlight RSA. Here I plot the Pearson correlation difference in brain to model alignment. Overall, we find that our LLM-trained RCNN outperformed the model that was trained with categories in most visual areas, with the exception of early visual areas, as you can see along the calcarine sulci here. And actually, the two models led to good predictions in these early visual areas. Finally, we tested state-of-the-art computational models reported over the years to be predicted of the ventral stream in the human ventral stream. So in this figure, the y-axis shows the representational alignment between the model and the brain data, and the x-axis shows the size of the training data set. And here we're talking about the visual images that are fed as input to these models for training. We tested unsupervised models, semi-supervised models, supervised models trained to predict the categories, supervised models trained to predict the scenes labels. And overall, all of these models predicted the brain data really well, but our LLM-trained model outperformed all these other models. So again, this is suggestive that having a good training objective can be really useful because our model had orders of magnitude less visual training data. So I'm a little short on time. I'll try to speed up. Next, we use voxel-wise encoding to try and predict the brain activity, the brain responses to the visual scenes using linear model applied to the embeddings of the scene captions. And to measure accuracy, we look at the correlation between the observed fMRI activity on a test set and the predicted brain activity with our linear encoding. Again, we observe a similar network of brain regions where the beta weights in response to a test set of images can be predicted with extremely high accuracy, even in single subjects, reaching all the way up to 0.86 in Pearson correlation. Again, this is using LLM embedding of scene captions, so linguistic models. So next we see, okay, we've learned the weights to predict brain responses. So if we write novel sentences like a man with a beard smiling at the camera or a view of the beautiful landscape, we should be able to recover known organizational principles that have been observed over the years in the neuroscience literature. And this is exactly what we see. We can replicate specific results, including in warm colors here, you see responses to face-like sentences, and in blue colors, responses to place-like sentences. And the white contour shows the regions of interest identified using the object category localizer of NSD. So to sum up, it's really fascinating that large language models can predict brain responses to visual scenes, and we think that's because their training regime allows them to capture the structure of the world. And before I go, I just want to talk about a novel NSD dataset that we're capturing, that we're recording, the NSD-EEG dataset. This is led by Peter Brotherwood. I'll be quick, but you can hear his talk on Wednesday describing this novel initiative. The data quality is excellent already, and it follows the same experimental design as NSD. We'll also have 3 Tesla fMRI response to a subset of the stimuli. To foreshadow Peter's results and Peter's tasks talk, using a linear encoding model similar to that that I've just talked to you about in fMRI, we can predict EEG amplitudes with high accuracy. So in this figure, in the y-axis, is the cosine similarity between predicted EEG amplitudes and observed EEG amplitudes. And this is using only a third of the data, so we're about 30% done in collecting the data. And I'll stop now before I steal time for the other speakers. But I would like to thank people who've been involved in this, especially Tim Kietzman and, most importantly, Adrien Dorig, without whom this would not have been possible. Of course, Kendrick and the creators of NSD, some of them are not on the slide, and the funders, and thank you for your attention.
We have time for one quick question while Thomas sets up the next talk. Any questions?
Yeah, great talk. Thank you so much. I'm just wondering, with these embeddings that take these labels, these descriptions to a semantic representation, I would expect to see brain regions more, let's say, towards the anterior temporal lobe, or it's a little surprising to get this strong correlation around the HMT, LOC. How do you interpret that?
So what I'm thinking is that because these large language models are trained on all the text that has been digitized, basically, since humans have started, making the text what they write digital, these models can capture what we describe, and we describe the world when we write. And so along the dimensions of these models, there's room to fit many of the components that may predict different visual areas. And I think this is illustrated by the result, where by training the linear model, we can recover known organizational principles in fMRI data. So the loadings on different dimensions enable to make these predictions. We have other results like this using sentences, including food items, and we replicate a recent result showing food selectivity in the visual brain. So I think it's basically capturing the structure of the world, and along the dimensions, there's room for different loadings that can be predictive of different levels of abstraction.
Okay, let's thank our speaker one more time. The next speaker is Thomas Knappen with a talk entitled Farewell to the Explore-Exploit Trade-off in Large-Scale Datasets.
First of all, I want to thank you all for being here and the organizers for inviting me, and welcome you to VSS 24. There we are. Yeah, so I'll show you some examples of how we've been using large-scale data sets in the lab in terms of exploring new things and also then validating those same findings and specifically combining across some large-scale data sets. I first want to highlight one thing that I'm really interested in as a scientific topic, which is how when we see the visual world, our experiences are very visceral. So I'm going to play you a movie that's actual stimulus material here, where if you look at this, you actually interpret what people are acting out, and you actually have bodily sensations as a result, right? So when we see these types of naturalistic stimuli, we vicariously engage in bodily sensations, and vision is the conduit for this type of sensation. And so, yeah, this is really the topic that I want to get into. You see the frown on his face there. It's probably going to make you feel a certain way now. So we want to go into how to find body-related processes in the visual brain, and I'm going to start with sensory topographies as a tool because it's my only tool at the moment. And so from vision, we know that we can actually map out higher order visual areas as a function of their relation to V1, right? Whenever you imagine or see a visual stimulus, it will produce a topographically specific pattern on the surface of V1 and in all of these high-level regions. And this sort of co-occurrence of spatially selective patterns allows us to map things out. So for example, when you are in your lazy chair at home and you're watching out into your study, the visual image that you see is actually being topographically projected to the surface of V1 here. The color coding is eccentricity. And if I would want to map out the retinotopy of V3, for example, I could, for every voxel in V3, look at what portion of V1, which patch in V1, best corresponds to the time course of my target voxel in V3, right? And if I do this for every voxel in V3, I am actually able to reconstitute the retinotopic map. In essence, what you're doing is you're sort of projecting the retinotopy of V1, the principal map of visual space, into the rest of the brain, in this case, the target region of V3. And so this sort of concept in terms of analysis strategy, we've used to map out, for example, during a retinotopic experiment, you can really find, for example, the boundary between V3 and V3AB or V4 based on just this analysis, just by referencing visual responses to V1. Now, how do I want to apply this to body-related processing? So our strong hypothesis here is that any body-related visual processing, and this is a subset of all possible body-related processing, should show itself as topographic connectivity to the primary somatosensory cortex and its homunculus. And so what we're going to do is we're going to fit S1 connective fields and try to somatotopically map high-level visual cortex. So in principle, what we're doing is the same as I just showed you on the previous slide, but then from S1, trying to project its homuncular map into the visual system. Here you see an example of us trying to do that in M1 and IPS, for example. Yeah. Now this project was spearheaded by Nick Hedger, my former postdoc, now faculty in the UK. He's not able to be here, regrettably. And he performed an analysis on the Human Connectome Project movie watching data set, which is awesome. It features 180 subjects, each scanned for a whole hour, watching all manner of different videos. And so the target here is that we are going to try to do this connective field modeling from simultaneously S1 and V1, so fit what you might call multisensory connective fields, and try to explain brain-wide responses using that technique. And what we're interested in here is trying to see whether we can sort of see homuncular structure in the visual system. And so what I'm going to do first here on this slide is to show you the entire brain result, and then zoom into high-level visual cortex. And the color map that I'll be using has sort of facial features, lips, etc. as red, and then goes over the homunculus, over S1, towards the feet, which are sort of blackish. Now, if you look at the entire brain, here are a couple of views. You see that we sort of replicate the canonical structure of the somatosensory system. From S1, you see the homunculus pop up in M1. There are replications also of like medial frontal somatosensory cortex there in all of the sort of topographic features that we know of the system. But I want to focus now on these intricate patterns of topography that are seemingly there in visual cortex. So what I'm going to do now is cut those out and show them specifically. So this is also overlaying some region of interest definitions, partly from NSD, partly from the Wang Atlas retinotopy. And what you're seeing here is that more or less the entire lateral and dorsal portions of the high-level visual system are tiled with these types of maps. And you can actually delineate, based on these gradient reversals, edges on high-level visual cortex, which I'm doing here. This creates a bunch of quadrilaterals that you can then project onto a rectangular surface, which allows us to then use this as a reference frame later on, so I'll get back to that. Now, on the surface of the cortex, these maps are highly correlated across the two hemispheres, and also if you split across movies and across subjects, you find that this structure is highly robust. So we want to try to interpret this type of structure. So we know that from older literature already that there's a lot of regions in specifically this type of high-level visual cortex that selectively respond to the presentation of human bodies. And this is a canonical finding. These regions, extrastriate and fusiform body areas, are prime examples. And our hypothesis is that from this homuncular organization that we get from our connectivity measures, we can actually predict a map of visual body part tuning. And so what we did is we fit single voxel body part encoding models on NSD. So we're moving from HCP movie watching to NSD. And what we did is we tallied the occurrence of specific body parts using key point detection AI in each of the COCO images, then allowing us to create a design matrix across images and across trial responses, and then just use ridge regression to fit a single voxel tuning curve in the dimension of body parts, if you will, for each voxel in the brain. And so now we're going to try and see if this sort of thing aligns with these putative body maps that I've shown you. And this is that sort of reference frame that I tried to build up from those quadrilaterals. On the top, you see how this sort of aligns with known visual regions. There's the extrastriate body area over here in the lateral visual system. And I accidentally clicked. So here you see the results of that PRF mapping type, encoding model type fit. And you see there's some structure there. It's strongest near this area PH and the EBA. And if we correlate these maps with one another, we see that there's a strong body part tuning correspondence. So coming from movie watching and the referencing to primary somatosensory cortex on the one hand, and visual content, the appearance of certain body parts in visual images. Now, this contrasts with a separate analysis we undertook highlighting spatial correspondence. So you could also say that, in a general ecological setting, you have spatial correspondence between body parts and retinotopic locations, so that heads are always in the top and feet are always in the bottom. And you can use HCP retinotopy to do this top-bottom distinction and then correlate that map with our homuncular map. And this type of result then complements this body part selectivity part. So we have a non-spatial body part selectivity mode of operation, perhaps, and a more spatially oriented correspondence there. So to conclude and start looking forward, I think what I think this shows is that this somatotopic organization, as referenced from S1, is one of those principal organizational motifs, such as retinotopy, such as object category selectivity, such as object size, et cetera, in high-level visual cortex. It also aligns really beautifully with the semantic maps we just saw. And what this somatotopic structure shows is that it actually allows us to predict a map of body part selectivity, visual body part selectivity, in this relatively unknown region PH on the one hand, and in other regions it corresponds to visual space tuning. And what I think this might point at is a set of different interfaces that allow vision and touch to interact and thereby generate vicarious experiences of things we see. And so this really highlights how you can tie together, and that's what I really like about this in terms of also how this symposium, right? We're able to now sort of bring together a couple of these really large-scale data sets and look at their overlap. And it really shows the power of this type of approach for vision science. And just as a more philosophical side note, if you imagine this as the space of possible vision science inferences and a single scientific inference that you could do as one of these points here, if you do a hypothesis testing type experiment, you're going to target a specific inference, right? And any sort of reuse of your data is going to be accidental. That's these points falling inside the dashed circles, right? But with these very large-scale fMRI data sets, what you get is an experiment that is not trying to test a single hypothesis. It actually tries to span this space. And then when you try to combine these different large data sets, you are able to capture an even larger portion of this inferential space, if you will. Now, I have 10 seconds or so. We're trying to go from consumers to producers of these types of data sets. And one of the things we're trying to do is acquire an NSD-style arrow of time data set with around 15 sessions per participant. These are richly annotated videos that we are presenting 4 times, twice played forward and twice played backwards, so that we can try to look at spatiotemporal type responses, things about how our brains extract causal information from these brief segments, right? So we are starting to acquire data. We have a couple of subjects in the pipeline, and this is our NSD style type D R squared, so it's on a par with NSD, although of course not the number of trials per subject. And we have an additional deep retinotopy data set that Martin Hebert's lab will be presenting tomorrow morning, which is deep retinotopy, sampling up to 12 sessions of PRF mapping type experiments. Thank you for your attention.
We have time for a question, perhaps, while the next speaker sets up.
Excellent talk. I have a question about your results. So I think there's the general consensus that there are 2 lines of higher category level visual areas, one in the ventral stream and one in the lateral stream, where the lateral stream is associated with dynamic stimuli and movies and affordances, whereas the ventral stream is associated with static images.
Yes.
How do you think about that literature and your results?
Well, so I think on a broad level, these results perfectly align with that, because we find hardly any of these types of things in the ventral system. So it's all dorsal and lateral, what we find. And I think this has to do with the ventral system just being like a static image processing type thing. Like that's why CNNs work so well there, I think. But yeah, that's an open question, so yeah.
Okay, let's thank our speaker one more time. Our next speaker is Jacob Prince, and his talk is entitled "Large Datasets: A Swiss Army Knife for Diverse Research Aims in Neuro AI."
All right, thank you, Kendrick. I took this picture exactly 5 years ago today, my first day at VSS. And since that day, one of the most energizing and productive things to happen in my research life has been getting involved with large-scale visual neuroimaging datasets and getting to know the community that has been developing and supporting these resources. And I want to speak about these datasets from the user perspective, as someone who is not involved in data collection. And in particular, I'd like to show how, for a wide range of users, these datasets can help form the foundation of a diverse research program encompassing approaches from neuroscience and AI. And I've come to think of these like a Swiss Army knife, a multi-purpose tool that can support many different kinds of projects. So here I'm going to tell you about 3 different research anecdotes, 3 different use cases for these datasets, each offering a particular lesson about their value. And the first story is about fMRI methods development. I first came across these datasets because I was interested in how category selective regions relate to one another and to the rest of high-level visual cortex. And here at VSS, I first met Mike Tarr, who suggested it could be useful to look at BOLD 5000, as well as NSD, which had just been collected around that time by Kendrick. But when I started poking around these datasets, I faced a confusion that will be familiar to some who have analyzed NSD, which is why are there so many different versions of the data, each reflecting different pre-processing choices? What's different between them, and why do these differences matter? I got an answer when I started looking at the reliability of the different data versions and realized whatever was happening under the hood had a really serious impact on data quality. And this was the effect of GLM Single, the optimized pre-processing pipeline that Kendrick had developed to tackle the signal estimation challenges of large data sets. And so with a group of collaborators, we worked to make this resource available to the broader community. And briefly, I'll just go through how GLM Single works under the hood. Using either MATLAB or Python, a user can provide a design matrix and fMRI time series, and the outputs are beta weights representing estimates of single trial response amplitudes. And the first optimization happening under the hood is the use of a library of hemodynamic response functions to identify the one that best matches each voxel. The second procedure is GLMD noise, which takes data from noisy parts of the brain and uses it to derive a set of noise regressors that we can include in the GLM to optimally improve the quality of the betas. And third, to actually estimate the final betas, GLM Single uses an efficient implementation of ridge regression, and this is a really effective approach for regularizing signal estimates in cases where the regressors are super close together in time, which is often an issue. And so, what's the impact on real data? Why does this matter? This is an example of a voxel actually from an auditory study, a subject in the music listening component of the study Forest dataset who heard 25 musical clips from 5 genres. And these are the betas you would get using a generic GLM approach without any optimization, but now using GLM Single, you get something like this. Reliability has increased substantially here, and this is not just a cherry-picked edge case. Here's another voxel from that subject, shown here with the pink star. And so anything above the diagonal here indicates improvement in reliability, and this is the trend we saw across subjects. And these trends were also similarly strong when looking in NSD and in Bold 5000. GLM Single has now been used in several other published studies, and we've actually released a new version of Bold 5000 with optimized signal estimates. And so this work shows how these large data sets can provide an ideal test bed for new methods innovation. I think a deeper lesson, too, is the value of focusing on reliability in our research efforts, because it's critical to quantify and understand the meaningful variance we're working with, and to keep pushing toward paradigms and processing tools that maximize the richness of our modeling targets. The second anecdote now I want to talk about is how these data sets can be used to help resolve long-standing debates in cognitive neuroscience. Category-selective regions for faces and bodies and scenes and words are classically thought to act as independent modules because lesioning or otherwise perturbing these areas tends to mostly impair recognition of the preferred categories. But we can't have a dedicated module for all the thousands of different kinds of things we recognize. So an alternative view is that these objects are all represented with distributed patterns of activity over large-scale populations of neurons, kind of like a barcode in the brain. And it's true that you can decode lots of information about many objects across the ventral stream, for example, decoding cars from chairs in the fusiform face area. But it's not clear how just the presence of that information actually relates to downstream behavior. So are these different views compatible or opposed? We gained new insight by studying a class of self-supervised neural network models that have a generic architecture with no specialized architectures or branches for different domains that learn from a rich image diet with the goal of representing each image distinctly from every other image with no labels. And even without category supervision, these models learn rich features that support accurate category readout. And interestingly to us, when probing these models with the same stimuli you'd use to define category selective regions, we found that they developed emergent category selectivity for all these domains. But do these units look anything like modules? We lesioned the face selective units, and we found that the biggest impairments for processing were for categories that tended to activate those units highly, mostly animate images with lots of faces in them. But when we lesioned the scene selective units, there was a completely different profile of deficits. Recognition of large objects was impaired, and outdoor scenes. And so, really importantly, over the full set of 1,000 categories, we found that the profiles of deficits from these different lesions were uncorrelated. And this double dissociation suggests that generic self-supervised learning led the model to develop something that looked like implicit modules for processing different kinds of content. But could these units actually predict the corresponding category selective regions in the brain? And here's where the large data sets provided a critical validation test. looking at the mean response of each brain region to over 500 stimuli from NSD, we found that the selective units were able to account for substantial structure in these rich brain response profiles. And as a point of comparison for FFA, we also looked at a model that was trained to identify thousands of different face identities and learned that it could explain far less structure in that region. And the same story held when we were looking at the representational geometries. So overall, these results show that generic self-supervised learning actually is yielding brain-aligned category selective features. And these results helped us propose a way to reconcile this long-standing theoretical tension between modular and distributed views. Our lesioning analyses support the modular idea that high responses signal a region's function, but we can see that signature without learning category-specialized tasks like face recognition. And our results also support the distributed viewpoint that a single large-scale population code can learn to tell all the objects apart, but that does not mean that both high and low responses matter equally for recognizing any one object. The high responses appear to be a much stronger signal of function here. And so these empirical puzzle pieces can be understood within a single model as a consequence of generic self-supervised learning. And a deeper lesson with this use case is about the importance of hypothesis-neutral, condition-rich sampling in our datasets. After many years of trying to study these regions separately with domain-specific image sets, that made it harder to actually understand how they might relate to one another. And the richness of NSD was critical to unlocking that link. The final anecdote I want to talk about is the neural network benchmarking at scale. And so what learning pressures actually underlie brain predictivity among these neural networks? This is work with Colin Conwell, and thanks to tremendous initiative on Colin's part, we developed a pipeline for efficiently using data from high-level visual cortex in NSD to measure the brain alignment of hundreds of neural network models. But instead of just searching for the most predictive model, we implemented what we call opportunistic experiments, comparisons between groups of models that are identical in all their inductive biases, except one, which we vary. And we found that advances that had really impacted the field of AI, like transformers, actually weren't giving greater brain prediction than ConvNets. We didn't see better prediction from just scaling up the training set, for example, to ImageNet 21K, and language alignment alone was not sufficient to drive up prediction, holding the training set constant. Instead, we found evidence that the most predictive models came from having training diets that were the most rich and diverse. And we see this as further evidence in favor of the primacy of visual experience in pressuring the learned representations that we measure in visual cortex. The value of this paradigm is also reflected in other efforts to study groups of large groups of AI models at scale, like the Algonauts Challenge. And these community-driven efforts, I think, have real potential to drive toward better models of neural representation and function, especially when we pool our data across modalities and across species. And I'll just cover a brief set of items, maybe a wish list for future data sets. It's important to note that none of the projects I just talked about even came close to using the full scope of NSD and Bolt 5000, and it's possible that we might benefit from having more trials per condition, perhaps at the expense of having so many conditions. In addition, we might think about balancing sets of naturalistic stimuli with smaller, more curated stimulus sets. And finally, these do involve a bit of code wrangling just to get the data loaded and make sure all the stimuli are in the right order, so we could benefit from having better prepackaged subsets of data to help ease the coding burden. In conclusion, I'll just say that these resources really reflect what I think is a great way to go about science with collaboration, open sharing of data, and I look forward to chatting with all of you and the rest of VSS, and maybe even to collaborating too. Thank you.
We have time for a couple questions.
Yeah, thank you for the talk. I have a questions related to experimental design. If I want to use DRAM single method for my experiment, how many repetitions of stimuli should I collect at least? Do these repetitions need to occur at the same run or same sessions, or actually it doesn't matter? Thank you.
Thanks for the question. How many repetitions are ideal for using GLM single? We found with NSD and Bolt 5000 that we could take advantage of the optimizations in GLM single, having a very few number of repetitions available within session, with NSD having 3, and in some sessions of Bull 5000 having only 2. But obviously more is better. So yeah, and there are also ways to take advantage of these tools, even with no repetitions available, and some groups have done that as well, but it just involves coming up with some clever heuristic settings of the hyperparameters. So happy to chat more about your specific use case. Yeah.
Any other quick question for Jacob? Okay, let's thank him one more time. We're running pretty good on time, but we have one last talk and then plenty of time for a nice discussion, hopefully. The last speaker is Ale Gifford, fellow organizer of the symposium. His talk is entitled Synthetic Large-Scale Visual Neural Datasets.
Thanks again to the VSS organizers. I will propose a research framework centered on synthetic large-scale neural datasets. That is, neural responses generated by computational models of the visual cortex, and argue how this framework can benefit vision science research. During the last hour, you saw many examples of the potential of large-scale visual neural datasets. But unfortunately, these datasets are expensive to create, they take a very long time to acquire, and are limited to the stimuli presented during data collection, which is in stark contrast with neural data simulated by computer models, which is cheap and quick to generate, and it's unrestricted to any stimulus. The good news is that we can leverage existing large-scale neural datasets to train computational models that will effortlessly generate neural responses for any amount and type of stimulus, so to facilitate and speed up research experiments. And I will now show you how we did this using encoding models. Encoding models are algorithms that take visual stimulus as input, like an image, and synthesize the corresponding neural response. And their prediction accuracy increases with more training data. For example, here you can see how the correlation between encoded and real EEG responses increases as a function of the amount of data used to train these encoding models. So because of this, we trained encoding models using the largest available data sets of both fMRI and EEG responses to naturalistic images. For fMRI, we trained feature-weighted receptive field encoding models using the natural scenes dataset. fMRI responses of 8 subjects to 73,000 natural scenes. And these models had higher prediction accuracy for early visual cortex, where they accounted for up to 65% of V1's explainable variance, and accounted for almost 40% of the explainable variance across the ventral stream. For EEG, we trained vision transformer-based encoding models of things EEG2, which are EEG responses of 10 subjects to over 16,000 images from the things database. And their prediction accuracy reached the noise ceiling level at around 100 milliseconds after stimulus onset, and remained close to noise ceiling level for the entire epoch. We then made these fMRI and EEG encoding models available as an open resource called the Neural Encoding Dataset, NET in short, and created a GitHub package so that you can use them to quickly and cheaply generate synthetic fMRI and EEG responses to any image of your choice, which is one line of code. To help you familiarize with this resource, NET also comes with tutorials and a data manual. And to facilitate research, NET also includes pre-generated synthetic fMRI and EEG responses for 150,000 naturalistic images coming from 3 of the most widely used image sets in vision neuroscience. And I will now provide an example of how we used the neural encoding dataset to create a new neural control-based algorithm. So neural control is a method used to find stimuli driving a target neural state of interest. Usually this target neural state is high response magnitude, and inspection of the controlling stimuli can be informative of functional properties, such as the visual features to which a given voxel or neuron are tuned to. For example, here, this hypothetical neural machinery is tuned to actress Deep Ullman. When it comes to fMRI, neural control has so far mostly been used to investigate functional properties of areas in isolation, although given that the brain is an interconnected system, functional properties of one area might be best understood in relation to other areas. And it only focuses on maximizing or minimizing univariate responses, whereas we know that visual information is also present in multivariate population patterns. We therefore introduced a new neural control method called relational neural control, which finds images that jointly control multiple areas by aligning or disentangling both their univariate and multivariate responses, therefore highlighting functional similarities and differences at different levels of visual information coding. And since the functional properties of early and mid-level visual area are less well understood, we applied relational neural control on the univariate and multivariate synthetic fMRI responses from the neural encoding dataset of areas V1 to V4. And I will now show you how this worked for the multivariate control case. So the goal is to find images that align or disentangle the multivariate responses between early and mid-level visual areas to highlight both functional similarities and differences. So we started by creating random image batches of 50 images from the 73,000 images from NSD. And we then fed these image batches to nets encoding models of two areas, say V1 and V4, and synthesized the corresponding neural responses. Since we wanted to compare the two areas, we had to bring their multivariate responses to the same format. And we did this by transforming them into representational similarity matrices, or RSEMs. We then compared the RSEMs of the two areas with a Pearson correlation, resulting in one correlation score for each image batch. To align the two areas, we selected the image batches with highest correlation scores, which contain images that are most similarly represented by the two areas. We finally fed these scores to a genetic algorithm optimization, which used them to generate new mutated image batches and repeated the same loop over many generations. Instead, to disentangle the two areas, we selected the image batches with lowest absolute correlation score, fed them to the genetic algorithm, and again looped over generations. And ultimately, the algorithm optimized to a single batch of images, which either best aligned or disentangled the multivariate responses between areas. We cross-validated this algorithm across nets in silico fMRI responses of different subjects and found that the aligning images resulted in correlation scores very close to ceiling, and that pairs of non-adjacent areas could be best decorrelated, suggesting that functional differences might increase the further away two areas are from each other. Next, we focus on the V1 and V4 comparison and visualize the corresponding images. The aligning images often contained empty regions, for example, the sky in the upper half, whereas the disentangling images did not. And since V1 and V4 are both retinotopic areas, alignment might be largely driven by both areas similarly encoding topological properties of image, such as the spatial location of the objects. Since we applied relational neural control on nets and silico fMRI responses to know whether these effects are real, we had to see whether they generalize on in vivo fMRI responses. We therefore recorded fMRI responses from independent subjects for the V1-V4 comparison, controlling images, and again found that the controlling images well aligned and disentangled the multivariate responses between V1 and V4. Relational neural control also well aligned and disentangled the univariate responses between areas, and if you're interested in this, you can come to my poster on Sunday morning. So together, these results provide new insight into the functional relationships between early and mid-level retinotopic areas. They validate relational neural control as a new method, and crucially also demonstrate that the neural encoding dataset is well suited for research purposes. And specifically, they validate the neural encoding dataset for method development and exploratory research, but its application might also extend to hypothesis testing, results validation, model development, and NeuroAI research. The advantage of synthetic large-scale neural datasets, such as NAT, is that they can easily be scaled up to neural responses for hundreds of thousands or even millions of images. This in silico generation of neural responses is resource efficient, saving enormous amounts of time and money, and can therefore greatly speed up research processes. However, this does not suggest that traditional data collection efforts should be eliminated. Instead, this framework proposes a virtuous cycle where fast-paced research is performed on in silico data, its findings are then validated on in vivo data, and the novel understanding coming from this validation in turn informs new fast-paced in silico experimentation. Of course, this new framework also comes with limitations. Above all, the limited prediction accuracy of encoding models and their limited ability to generalize out of distribution. However, these limitations are overcomable. The prediction accuracy of encoding models will improve also thanks to initiatives such as the AlgoNudge project. Oh no, sorry about that. A challenge where participants cooperate and compete in building the best encoding models of brain responses. And I'm happy to announce that the 2025 edition of the challenge will open in fall and will consist in building encoding models of fMRI responses to naturalistic movies from the C-Neuromod dataset. Over 60 hours of fMRI recordings per subject while they watched episodes from Friends. And out of distribution, generalization might improve by training encoding models on a denser and wider sampling of the visual space, for example, by using multiple datasets coming from different visual distributions. As for the future, NET is designed to be scalable and can rapidly expand, for example, to new data types, such as ECOG or animal data, and also to new modalities such as videos, language, and audition. And to make this possible, we strongly welcome any ideas and collaboration. So to conclude, I would like to thank my collaborators and funding sources, and we strongly appreciate receiving your thoughts, ideas, criticisms, and also collaboration so to improve and expand the neural encoding dataset. For this, you can approach me at any time during the conference, you can come to my poster Sunday morning, or also drop me an email. We're looking forward to your feedback.
We have time for some questions for Ale. Okay.
No questions.
So we'll transition over to the Q&A and discussion, which I will moderate. Can you? Yeah, that's fine. So we have a few things to go over. So at the very beginning, hopefully you were here, we had this very quick survey of basically questions and issues that you, as the audience, want to know about. I'm going to go over that in a minute. I know that was a lot thrown at you, 6 very rapid talks. Hopefully you enjoyed them. This is fun. So just a few QR codes. You can just take a picture and you can fill these out later. You saw links to these earlier, I believe. One of the themes here is if you like or might like engaging in this type of research, it's a very different model of doing science. It actually is it's a cross-lab thing. There are dataset creators, dataset users, collaborations that form amongst these various groups. And this is, I guess, requiring a new way of doing science. And we set up a Discord server. There's not much traffic on it because I'm not sure we know how to navigate this new world. But it does exist. And if you take a picture of this, you can join. And it may change how we as a field do science. Another link you might want is this. This is a longer formal survey. We do want to know what you think. Elena mentioned this in her talk, but it's still open, the survey. It's coming from NSD, but we do want, NSD is not the only data set, and we want to know what the community wants and feels about other neuroimaging large-scale data sets out there. So this is it's a 5-minute survey, but please take this if you're interested. So to kick off the discussion and panel discussion Q&A, I just want to do something a little hokey, but I think it's useful. The room is thinned a bit, but now you're in this room, you can't leave. So I want you to do this. If your birth year is even, turn to your left. And if your birth year is odd, turn to your right. So we want to form little small groups. We'll just take 5 minutes to get you talking. You've received a lot, but you need to transmit a little to your neighbors. And I just have 2 simple questions for you for the next 5 minutes. And I know this doesn't actually work. There might be chains of 8, but this is just a starting point of forming small groups. I want you to think about and discuss these simple 2 questions of the 6 talks you heard. What's something you found interesting, you actually liked? And the second question is, what's something that annoyed you? And both are really important questions, so please discuss both. We'll just do this for 5 minutes. similar to what?
To where the results of this was exploratory analyses. Maybe not. She had also a graph of your question for survey.
Cross survey.
Yeah.
We'll go for 3 more minutes. So wrap up your conversation, 3 minutes.
More subject. More community. No, more conditions is less.
Okay, let's reconvene. Hi, everyone. Well, this is a good sign that the conversation is still going, but we want to share thoughts across groups. We have plenty of time, by the way. We have half an hour, which is a great job on my part. No, I'm just kidding. So yeah, we have plenty of time to go over the survey results and actually ask the panel get the panel some input. But right now, since it's fresh in your mind, any small little groups want to share us some burning thoughts on what annoyed them or they pleased them? I can't see. Raise your hand.
Hi, we had a great discussion here. I do neuroimaging with detailed design, and my colleague here does physiological recording. So we were both very impressed by the quality of data and the amount of that can be collected, which cannot be done with a small research group. On the other hand, one of my little reaction was this is like, in some way, like a giant fishing expedition, right? You don't have an idea. Like usually, traditionally, research is done by forming a hypothesis and then make predictions that design the stimuli exactly tease apart the relevant dimension, which maximize the relevant dimension you're trying to study. Here, you have everything all at once. It's very complex. I mean, if you have, that's the one bit that's sort of different. I guess could be my training bias. I come from a tradition where you have to have something first before your advisor allows you to do anything, right? So here it's a reverse. You collect data, then hopefully somebody will form hypothesis to find something interesting. But then data is super complex. So sometimes you might get lucky, but sometimes you might not. So I guess that's what we're thinking.
Anyone want to react to that? Panel or others? Is that a hand? Or?
You have a hand right in front of you,
Yeah, so I agree with this, right? There's a certain power in hypothesis-based experiments that you just cannot do with such a wide sampling. So I think like the two approaches go hand in hand, right? Synergistically, you could explore on large data sets, formulate a hypothesis, and then very, very specifically, informed by the large data sets, go deep into the hypothesis testing sort of paradigm. So I don't see them as conflicting, but like mutually reinforcing, really, yeah.
Any commentary on that, Marissa?
Thanks. It was a great symposium, first of all. And I think we were saying we see obviously the virtue of this approach, which is great. But very much in line with that, I think the fact that for me, I would like to see sometimes maybe fewer subjects, but doing a task, right? Like, how do we understand more about the process too? So I think this is great, for the mapping. And again, I agree that they can be complementary. But I think at the same time, we don't lose sight of the other part that could be having maybe fewer observers, but lots of repetitions and doing different tasks that we could actually model and tell you more about the processing. And I think we both agreed on that. You want to add anything or no?
I agree with that. Okay, let's switch to another. So in these little mini discussions, any group had something funny or positive or negative to share with the larger group here?
Okay, I want to say first that I'm a huge fan of large data sets. So, but a point of risk that I sometimes see is that the great thing is that we can all work on these data sets, right? And we can all write papers about them. But then also sometimes findings get presented as replications, even though they are in the same data. So should we be careful about when we say, okay, this is, we have this very robust finding, but actually it's just the same result, just measured with a slightly different method or slightly different encoding model or whatever.
Any comments on that issue, other than Thomas? No, just kidding.
No, I think this is the whole Feynman thing, right? You want to separate your hypothesis generation process from your testing process, right? And as soon as those two get muddled together, you start to, get circular reasoning as a field. And so I think, as we use these data sets as a field, they will inherently become stale, right? Because findings from them will diffuse into not just literature, but also in the community's thinking, right? And that will be overfitting, right? So yeah. I agree.
What, for example, Jan was showing is that he was using a single data set, but the results were robust and validating across all of the subjects, and the analyses were performed independently on all of the subjects. So unless you assume for any reason that there is a specific bias due to the way the data set was collected that is propagated to all of the subjects that lead to all of them showing the result, I think that also showing that this generalizes doesn't necessarily require, I think you can, to a certain extent, trust the effect, even though it has not been shown in other data sets because it generalizes across the population.
I just want to respond to that with the cautionary tale of these people are very committed to show up once a week, every year to be scanned. So I think there's some value in the fact that this might not generalize to a broader population. So if you have questions about population variability, then these data sets might just not be the right type of data for you.
But in that case, if it doesn't generalize, it's not because necessarily their brains work differently, but maybe because they provide better signal out of less head motion or whatever. Or do you think that because they're more motivated, then also they neurobiologically function differently?
I would say that people that live close to the scanning facility might come from a certain demographic, and I don't know, maybe that's very homogeneous, and I think you should think about that.
Okay, Jan is, okay.
Okay, I think there's a key point here that when you release a large data set like this publicly, you do have the risk that two labs come up with a similar idea, and that one lab might beat the other lab in publishing the results from this idea. And then what do you do if you're the other lab that was slower in generating this finding? Do you publish it? Do you explicitly mention, oh, this has been done before, but we don't want to waste our effort? We worked really hard on getting this result. So I think it's critical as a field to think about what kind of behaviors or principles that we want to adhere to when these situations happen. And it will happen, and it will happen even more the more large-scale data sets we collect. So I think that was a very good point, and we need to reflect on this as a field, and we need to think about more collaborative ways of investigating these data sets.
Okay, we're going to switch gears a little bit. We can come back to the free-form contributions. These are the survey results that was conducted in this room over the last hour and a half. I found a few of these actually quite interesting. So the majority of the responses say, no, I have not used these data sets. Totally expected. VSS is a very large topical landscape. And it is what it is, but you can see the distribution here. The next question was, what are these things good for? Hypothesis testing, exploratory analyses, training models, replication, developing methods. I would say it's pretty well distributed, which maybe not very surprising, but great. These are, as Jacob said, Swiss Army knives. Many, many, many possible applications. Next question. And these are the hard ones, like should you do this or that? Basically everyone wants more, but let's see what people prioritize. Do you want more naturalistic stimuli or more controlled ones, old school, or do you have no opinion? Looks like naturalism wins, at least in this room, with the blue much bigger than the red. I found that interesting. I found this even more surprising. We're at VSS, and people want multimodal data sets, vision, audition, language, or just pure vision. That would be a tiny slice of the pie here, red, and orange is no opinion. Next question. I found this one funny. Should we have more subjects, individual differences, diversity, demographics, etc.? NSD chose 8. 8 is not really a super well thought out number, but it's more than 2. Or more conditions like images or movies or what have you, or more repeats. So higher signal to noise per distinct stimulus. It's pretty much balanced, I would say. So no matter what you do, someone will be unhappy. All right, how to measure the brain. What's most valuable for these types of data sets? Pretty close between fMRI and EEG, MEG, which are probably the dominant ways, at least in humans. I guess EFIS was listed here. We didn't really say humans only, so non-humanid primates and other animals could, and there are efforts in the animal world, certainly for very large data sets, but very different type of data. And looks like behavior, people like behavior, I like behavior. So yeah, there's a vote for make behavior part of it. And if you remember, none of the 6 of the talks really talked about results about behavior, I think. The first talk, Elena's does mention, we want to collect behavior, that's the focus of an upcoming data set. But thus far, and we're just mining these data sets, we're picking low-hanging fruit, but currently behavior has been given the short end of the stick. And then finally, which is kind of the panel discussion, and we still have time if this is still of interest to you, here's what the community wanted to hear some thoughts on. And actually, can I have the speakers just stand up so we can enter that stage of the symposium here? Let's just start with the most highly rated question that the audience wants to hear, which is, let's talk about this whole naturalistic versus controlled divide, and let's limit the scope here. Of course, this is a topic you can talk about ad nauseum, and there's no real right answer, but remember, this is about these types of data sets, these very expensive, very onerous, very challenging neural sampling data sets. In the next 1, 2, 3, 5, 10 years, like, where should we put our money? So I'll ask the panel, anyone want to venture some thoughts on that? Well, I guess we already heard ongoing efforts. We know where Wanmock lies.
Well, I mean, I already talked about my opinion a little bit in my talk, so I'll advocate naturalistic, not just naturalistic stimuli, but whole naturalistic behaviors. Because, I talked about the reductionist approach in my talk. So our hope is that when we dissect this gigantic, complex cognitive processes into really elementary ones, then our hope is that the essence is going to be still preserved in that elementary functions. But then it may not, right? So as Yauda already talked about, the hypothesis-driven versus data-driven approach kind of a little bit. I totally acknowledge that this is a gigantic fishing expeditions, but I think it has a real value in it. Because if we are limited by our hypothesis space that we can think of, then what if it's just like limited and then totally miss the really important landscape somewhere over there, right? Then how can we actually go over there even with just our hypothesis space? So that's my view. So why we need this naturalistic approach. And the naturalistic behavior, not just perception. Our perception is not, I think, the perception per se, right? So our perception is needed to make an action in the environment, the useful actions for the agent, right? So in that case, I think perception should be considered as a more of a bigger context of perception, action, and other cognition in an integrative fashion. So that's why, so I advocate the naturalistic approach here. One more thing about more repeats, that part. So you may probably be concerned that there's no repeats in the naturalistic stimuli, naturalistic paradigm. But then one thing you can think of is that we can think about this repeats at a more abstract level. So for example, in my task, so there's no exact repeat, but it's the same in our everyday life. There's no exact repeat. But then if I think about, the sheep attacked by a fox, every time when it's attacked by a fox, it could be different, right? At a different angle or a different map, and the whole thing is different. But then in its sense, it's something, you are at the risk of losing your reward. It's the same thing. So maybe you can think about repeats in the naturalistic paradigm in that way a little bit.
I have a question then for you, Wenmok. Because you're the naturalistic person. Okay. I see the benefits, obviously, of naturalistic stimulation and experiments. I'm all for it. But I also see the reasons why people might want to have more control experiments. And even if you do not have a reductionist approach, or you don't go with that, still, one reason why you might want it to be more control is to reduce complexity. Because if you do have data which is too complex to model, then how do you end up modeling it? So and I think that this, throughout the decades, with scientific progress and method development especially, I think, then you can start modeling and using more and more complex data. But then my question to you, since you advocate let's go all naturalistic, do you think that we should go all in? Or do you think that we should do it step by step, such that the data set which we collect are also matched by the methods which we have, such that we can actually use them and not them being there without us being able to do anything with them? So what is best in terms of investing money, step by step or all in?
Well, it's hard to give binary answers for that question, I think. Okay, so I'm also, I'm also from a tradition of vision science. I do love controlled stimuli. I'm a big fan of, for example, random motion and the Gabor patches and everything. But then the thing is that, so the reason that I kind of converted to naturalistic paradigm over the course of years is that, it's actually, my stimuli doesn't contain the one that I want to look at. So that was the main reason. But you're right. So there are approaches that you can make the gradual steps toward it. But I think other people, probably, not me, other people would certainly do that approach. So we can be complementary. And also, I'd like to say that the naturalistic paradigms will give you some ideas that you might have missed in the past. But then that's not the end. I think if you got clues from these naturalistic paradigms or naturalistic stimuli, you can actually design more controlled or focused experiments, follow up on whatever you're finding. I think that in that way, it can be complementary, I think.
Can I respond? This feels like a nature-nurture question, and I feel like both are important, and we as the community should rally around topics that we find interesting and what data sets are missing. So if that is artificial, then we should make artificial data sets. If it's naturalistic, then, or like a combination of two. I think Thomas' point of overlapping space in the inference, like in the scientific inferences that we make, is important. That data sets can be used to translate to one another by having a common set of stimuli.
Hello. I'll step in a little bit and let's switch to a different topic, but I will have 2 cents. Okay.
I'm sorry, I just wanted to make a comment here because I think the interesting thing for me is that 42% said neutral or no opinion, and quite frankly, I was one of those because obviously I want more naturalistic, but I also want more control, and I didn't like the binary, the mutual exclusive choice. So I think it would be interesting to think what is behind those neutral no opinion.
It's interesting. I mean, if you just take who's the winner here, it's actually neutral.
That's what I'm saying, but it doesn't mean like neutral. It just means that I think many of us think we need both, right? And I was not willing to say one like controls, implying that I'm not interested in naturalistic or the other way around.
Yeah, I think that's an interesting point. Let me just chime in myself here. But if you read this at face value, this looks like a vote of confidence for OneMux, because it is multimodal and naturalistic, and that is the largest piece of the pie here.
Just add one. Yeah, so I think whether or not we push toward more naturalistic or more controlled, another thing to consider is that we also need to interrogate the methods we're using to get to our prediction outcomes. In a lot of the talks you saw like neural network models being compared to these data sets, and that's a very common paradigm. But for example, using RSA or encoding models, I think there needs to be complementary work to interrogate the assumptions of those methods. If our goal is to understand brain function, these data sets are so appealing for representational analyses, but it's not always the case that those analyses directly tell you from like a prediction, that the functional thing is there too. Just to give a concrete example, like in an encoding model, there's no guarantee that the features you're actually choosing to map onto the brain are playing the same functional role in the model as the brain region is in the neural system. So I think we need complementary focus on the linking assumptions as well.
Great. Let's switch to another question here. Although I did want to, I think that's a great point. It's like the data outstrips our methods or even theory. Arguably, you shouldn't collect a data set if no one knows how to analyze it. So it's kind of, you collect the data that the field needs at that moment, and hence you should ask the field what they think they need, is my opinion. Let's switch to this. I think we're okay on time. The second most rated question were the risks, limitations, and dangers. So I'll give the panel a chance to weigh in on what they see some risks are.
I mean, small-scale data sets are also important. And the risk, one of the major risks that I see is, what if funding bodies suddenly say, oh, why do you want to have money to collect this small-scale data set? Just use NSD, just use BOLD 5000. And without really understanding that the questions I'm interested in may not be covered by the sort of theoretical landscape of these large-scale data sets. And so there is a risk, if everyone today becomes obsessed and only starts collecting large-scale data sets, well, what's left, for the money pie, what's left for those smaller labs maybe that can collect small-scale data sets that perhaps tailor, or ask questions, specific questions that are not covered by the large-scale data sets. So I think this is important to reflect upon, to think about, how do we want to shape this? I think we want to continue collecting both large-scale and small-scale data sets.
Miriam?
Doing realistic, naturalistic for a while. I can say one of the challenges I've been facing, and I think, Womak, you probably will appreciate this too, that as soon as you turn into naturalistic, realistic, natural, you also face a lot of multicollinearity in your experiments. So stimulus, movement, attention, all of those are going to vary together, and it becomes almost impossible to disentangle. And to be honest, I'm not sure even controlled experiments can actually realistically disentangle those. So I think to be honest, I somehow in a way sometimes embrace the multicollinearity and then maybe think about future experiments to somehow disentangle them. But it is a challenge when you turn into naturalistic, and I don't know how to even think about the problem sometimes to get around that issue.
Any comments on this thread here? Right, I don't know your name. Can you run?
Thank you for the talk. Yeah, I'm Ken from Kyoto University, Kamitani Lab, and then I'm the decoding person. So yeah, I'm very interested in the RSA and encoding analysis. And then, so what's the risk of the limitation of the large scale? My comment is that, so naturally, a naturalistic stimulus is very good to analyzing. So I totally agree using the naturalistic approach. But on the other hand, we also perceive the control stimulus too. So even if you are focusing on the two naturalistics, we might forget the basic things. So we also perceive the control stimuli too. So I recommend that the large-scale data sets also contain the control stimuli, and then we can test such out of generalization performance or such another hypothesis testings easily. Yeah. So sorry for my poor English, but thank you for listening. Yeah, that's the comment. Yeah.
Yeah, so what I was wondering is that, so to ask what are the risk and limitations and what type of data should you collect, I have a question for the behavioral part, because Thomas mentioned that he was getting more into visceral responses, those types of things. Are you also thinking about measuring more of those responses in conjunction with your data? Because, like, for example, skin conductance, those types of things, to enrich the data that are there with multiple different quantitative measurements of physiology or physiological behavior.
Yes. No, I mean, I would love to have that, because I think we are neglecting a huge part of the types of behaviors that we are. If you look at this might be a segue, but if you look at the stringer type findings that we've seen, right, or the sort of twitch-like behaviors are causing a lot of brain responses, right? This type of, things like blood pressure, all that visceral stuff we are totally neglecting. And I think, there's a lot in there, right? We are not just CNNs, right? We are bodies engaged in the world, and that has these responses, or that evokes these responses. So we should be measuring that. The last I heard, it's pretty expensive.
Yeah, to add to that, there's also infinite behavioral data you can get from subjects, and it's expensive. I think there's an effort, I don't remember who or what, but an effort where they asked the subjects to essentially caption what they saw. Tell us what you saw, which adds a whole nother dimension, obviously, because it is from the subjects themselves as opposed to Mechanical Turk, and that's a whole nother world, obviously, of rich behavioral measures that you could try to mine. We have like a few last minutes, but we sort of hit a lot of these topics. I guess the third highly most rated was what types of LSVNDs are missing. So anyone want to end with like, what do they really want to show up on their computer in the next few years?
And I wouldn't like to directly respond your point, but what I would like to see from this community is, so from the point of view of a user, the problem with this is that you have huge amount of data, you need huge amount of resources and capacities. And I find it great that you collect this data, but I wonder if there is already maybe like some projects of analyzing data together. So instead of just collecting and then leave it to the community, also just, unite your force to ask questions all together.
Sorry, do you mean like collaborative, help me analyze this data because I don't have thousands of GPUs? Like, yeah, so this is kind of the reason we made this little Discord server.
Not only in the sense of like pre-processing or this kind, but really like just asking questions directly, because you're making like big data sets with like broad questions that are possible, but maybe, some people want to unite on a particular question and not just like on the data analysis at the beginning, but also up to the end.
Yeah, great point. Again, a topic that requires discussion amongst everyone with different skills and expertise and interests. And that's kind of why, well, we'll see if it works, this Discord server, whether it will spawn interesting projects. I think that's all the time we have. Thank you for attending and hope you enjoyed this.